Ladies and gentlemen, before the presentation of the fourth and the fifth part begin, I would like to introduce the presenter, Dr. Teh Chin Ho. He graduates with PhD in pharmaceutical chemistry from the University of Saint Malaysia. He joined Bruker Malaysia as an NMR application scientist. He supports the Southeast Asia region and assists customers, especially those new in the neutral products plant metabolomics fields. He expertise in with the different latest software solution of pure compounds and mixer to drive many possible NMR applications into the more accessible and better. He is part of the NMR methods and application development. Today, Dr. Teh Chin Ho give us talk about two main topics. The first topic is NMR applications in chemistry, material science, polymers, microbiology, and biology an overview. In this part, the audience will understand why scientists often choose NMR as it offers a variety of methods for research and quality control in biology, chemistry, physics, medicines, and material science. The range of application is broad, including materials, environmental and food science, medical and pharmaceutical research, and more. Whether for routine analysis of groundbreaking research, even small laboratories can now quickly adapt NMR in-house for definitive answers to molecular questions daily. And the second topic, Dr. Teh Chin Ho will talk about qualitative NMR or qNMR with targeted and non-targeted metabolomics with actual samples. The presenter will tell you how you can integrate multivariate statistics to NMR data collected on mixture samples. Non-experts could easily visualize hidden information within the complex NMR chemical fingerprint. Metabolomic fingerprinting has become a powerful tool for untargeted analysis of mixture samples and is gaining higher popularity, especially in food science, biomedical, and biotechnology. Moreover, the presenter will prove that the concept is applied to energy drink samples purchased from Thailand grocery stores, and NMR measurements were conducted in the Srinakarin Virod University NMR 500 MHz system. Untargeted analysis, namely principal component analysis and SIMCAL on these data indicate that a transparent clustering mathematical model could be easily achieved using Azure NMR Bruker analysis software. Without further ado, I would like to welcome the presenter, Dr. Teh Chin Ho, to deliver the presentation.
Thank you very much. So I would like to share the screen first. So I think you can see the screen sharing.
Yes, sure. Okay. Thank you.
Thank you. Yeah. So first of all, I would like to thank the organizing committee for inviting me to give a talk for today. So the first the talk I will cover the NMR applications in chemistry, material science, polymer, microbiology, and biology. So the applications of NMR actually is very diverse and wide. So it covers from the pure sample, which is the classical applications of NMR involving the small molecule samples, and also the macromolecular structure, so which is related to the protein. So for the classical applications, so this sample also have to be purified, and until currently the technology has been applied to analyze the mixture samples, so which is most of the times are small molecule analysis. And of course, this area NMR applications also can be applied to study the material samples that such as polymer. So I will cover several topics for this session. So first, these applications related to the chemistry, then I will move on to the material science and biology. So know that in the classical applications of NMR, so the sample can be obtained from the natural resources. So usually the scientists need to perform the several steps of the isolations and the separations until they obtain the pure material. And from this pure material, so the sample can be characterized by several techniques. So such as IR, mass spec, the X-ray, or another method is the NMR. So actually NMR is one considered a classical method in the medicinal plant chemistry. So this is the articles from Professor Tu Yu, so who was awarded the Nobel Prize in the medicine in the medicines. So she isolated the compound, so that is obtained from the plants called the Artemisia annua. So the compound name is the artemisinin. So in her earlier publication, so she used the NMR as one of the spectroscopy data to support the compound characterizations. So since then, there are many review papers talking about the applications of NMR, especially related to the characterizations and the identifications of the natural products. So when you look at the NMR result, so if you have the samples that have been purified and have been isolated, so the sample subjected to the NMR spectrometer, so you're obtaining the NMR data. So this NMR data, usually these are the fingerprints of that respective compound. So when you compare the NMR result, so they are highly compound specific. So even though there is a slight change in the structure, part of the structure, it will change the fingerprint entirely. So actually NMR is also, we say that it is a chemical fingerprint of the compound. So this chemical fingerprint, not only compound specific, but when you take a look, so this the same sample that measure the different time point. When you look at the NMR data, basically they are identical. So it means that the NMR result also highly reproducible in this sense. So not just a highly compound specific, but also highly reproducible. So with this, the chemists or the natural product chemists or the phytochemists can use the NMR as a tool to identify and to characterize the natural products in general. So related to the medicinal chemists or the synthetic chemists, so they also use the NMR quite frequently. So when they're doing the synthesis, come from, let's say, the organic synthesis, or it can come from the biosynthesis. So usually it required a series of steps. Start from the starting material, which is commonly known as a precursor, and to the products. So very often it may go through several steps. So which are the intermediate steps. So if this process, okay, for the synthesis process, synthesis steps sometimes may take weeks or months. So it is important to validate the samples, or if we call in between the intermediates. So otherwise, if there's something change in between the unknown factor, so we are not able to obtain our desired target or desired product. So they can be very troublesome to the synthetic chemists after spending several months. So in the normal practice, so usually before they perform the synthesis, so they will try to verify the precursor, and before they verify the products, so usually the synthetic chemists will try to verify the intermediates as much as possible, because by performing the verifications on these individual steps or the synthesis steps, so they can ensure that the reactions goes according to what they want. So that is the applications of NMR related to the analytical chemists or the synthetic chemists. So next, I will move on to the applications of NMR related to the material science and the polymer. So there are several applications related to the materials, especially the polymer sample. So when you take a look, okay, so the applications of the NMR related to the polymer, so make use of the NMR superconducting magnet range between 400 to 600 megahertz. Because we are studying the polymer sample, and the polymer sample consists of a lot of repeating unit, so we do not need the ultra high field system to study the NMR. So typically between 4 to 600 megahertz is already sufficient for the polymer analysis. So this is probably one of the earliest publications that mentions about the applications of NMR on the polymer analysis. And there are many chapters or the reviews talking about how the NMR can be applied to the polymer sample. So this is another example using one of the experiments, so which is we call it the carbon-13 NMR, to study the polymer sample that is come from the ethylene base sample. So this is one example. And another example is the NMR also can be applied to study the to study the molecule by applying the different techniques. So for example, the two-dimensional NMR technique to study the polymer. So this is another example, talk about the 2D NMR to analyze the polymer. So this against in this context, so the NMR serve as the tools to characterize the polymer in general. So of course, the NMR also can be used to study the change, okay, or the branch change that related to the sample polymer. So in this case, this is the polyolefins. So there are many examples talking about also the applications of the NMR to the polymer identification, especially related to the molecular weight. So this is the use of the pulse field gradient, which is the NMR technique, so that able to separate the polymer size according to the molecular weight. So diffusion polymer is another example, so which is very similar to the gradient spectroscopy for the polymer separations based on the molecular weight. So the very recent papers talking about the applications of the NMR related to the macromolecular size, which is also known as the material size. So this is again one good review paper. So if you want to know a little bit more, so please take a look, okay, on this paper. So of course, the NMR not just provide us the qualitative analysis, especially for the characterizations of the polymer sample, but also can tell us about the contents that related to the polymer. So actually the quantifications using the NMR also long established. So when you take a look, one of the example here showing that we can use some parameters from the NMR result to perform the calculations and the analysis on the monomer content. So again, this is very one of the very powerful tools. So we are not just knowing the characterizations of the polymer, but we also know the chemical constituent or the contents of the polymer based on the example here. So this is one of the example that talk about the propylene 1-butene copolymer repeating unit. So next, I will move on to the third, another example, so which is the applications of NMR related to the biology and the microbiology. So the biology and microbiology actually can be traced back to the one of the classical NMR applications, which is related to the macromolecule. So this macromolecule related to the biological fields, okay, the sample can be obtained either from the proteins, or you can measure the nucleic acid such as the RNA or the DNA, and then also part of the applications that related to this macromolecular structure is the protein ligands interactions, which is considered useful steps in the drug discovery. So I will start, okay. So the nucleic acid research using the NMR also been a long time ago. So when you take a look into the applications that related to the protein as well, so it's also a long time. Okay, so in the use of the NMR in the protein. So this is the one example showing that the 1984, so the first protein structure determined by NMR spectroscopy, and then the 1990s, so the first RNA structure have been determined by the NMR spectroscopy. Since then, there are many structure have been fully established by the NMR spectroscopy together with the RNA structure and the DNA structure. So when you look at these, the applications that are related to the macromolecules, so the sample have to be purified. Okay, so the protein sample or the RNA sample have to be purified. So while we go through these purification steps, okay, so probably in the protein sample that are related to the protein NMR, okay, in this context, so we need to purify the protein, and we also need to very often for the protein sample, in order to be well studied by NMR, so we need to label the protein sample. So this is the very special techniques, and this is also a very important technique that related to the protein that it has been studied by the NMR thoroughly. So this labeling technique will depends on the how big the proteins that we are studying, and then we can determine the protein labeling scheme. So why in the protein NMR or the macromolecular biology, so we need to label the protein? So the main reason is the sensitivity. So if you just take a look at the natural abundance, so the most favorite nuclide or the detection nuclide in NMR spectroscopy is 1H. So which is a protein, or which is also known as a proton or the hydrogen. And the second nuclide, also very important to the NMR spectroscopy, is the carbon 13 nuclide. And the third nuclide is the favorite nuclides from NMR is the nitrogen, the 15 nuclide. But when you take a look at these, the compositions and the natural abundance, so the percentage are very low. So this explains why the nuclide carbon 13 and the nuclide nitrogen 15s are very insensitive, although they are favorite nuclide, but they are insensitive. So as a result, in order to improve the sensitivity, so the biologists need to perform the isotopic label or the isotope enriched to the protein sample to improve the sensitivity, so that they can use the NMR technique to study the structure or the dynamics of the macromolecules. So as I said just now, so the labeling proteins will depends on the protein size. So if you're looking at a very small protein, so you just need a unilabel single label sample, so which is commonly labeled with the nitrogen 15. So if the protein size comes from close to 10 kilodaltons to 20 kilodaltons, so probably you need double label samples in order to improve the sensitivity, and you can apply the triple resonance experiments to study the protein structure. And if the size of the protein larger than 20 kilodalton, probably you need a triple label sample. So for example, the nitrogen 15, carbon 13, and the deuterium label sample, so that you can expand the TROC experiment for the analysis. So very often beside this labeling technique that been applied to the protein sample, so in the protein macromolecular study, so usually also using the ultra high field spectrometer. So very often that the scientists need to consider the gigahertz system so that they can get the better resolutions on the NMR result. So one of the examples that I want to point out is the NMR applications related to the protein and the RNA that related to the SARS-CoV-2 virus that caused the COVID disease. So in this research, so actually the NMR has been applied to study the RNA from the COVID-19 from the virus, the SARS-CoV-2 virus, and then also the protein sample that obtained from the SARS-CoV-2 virus. So these two samples have been well established and well studied by the NMR spectroscopy. So from the COVID-19 project, NMR project, so there are many scientists, researchers that are working on this SARS-CoV-2 virus. So the groups that targeting the RNA contents that related to this SARS-CoV-2 virus. So this is the example, the NMR spectrums that related to this secondary structures of the RNA, so that using the 1 GHz system. And there are also publications that related to this RNA of the virus, okay, SARS-CoV-2 using the NMR spectroscopy. So the consortium also study very well the structure, protein structure that come or isolated from the protein from the virus. So these protein structures can be linked to the non-structural protein and also the structural protein such as the spike structures, okay, and also the accessory accessory proteins that related to the virus. So this is example the 2D NMR that perform on the non-structural proteins from isolated from the SARS-CoV-2 virus using 1.2 GHz NMR system. So this is another example. So which is also the non-structural protein. So by analyze the protein sample after performing the labeling, okay, so the scientists can understand the protein structure very well, and after that they can use this information try to develop some antiviral drug based on the drug discovery campaign. Yeah. So this screening target is the next steps after they understand the secondary structure of the RNA from the virus. So that is related to the binding ligands interactions. So these ligands binding interactions can involve the ligands and the protein, or the ligands and the RNA. And that is a very important approach in the drug discovery process. So in the drug discovery process, first of all, we want to know the scientists want to discover the new drug. So this actually one of the example is called the vemurafenib. So this is a new drug that obtains from this NMR approach, okay, that is derived from this drug ligand fragment based screening. So this drugs actually starts from the very simple fragments. So the developments of the drug start from the fragments. So from the fragments, so the scientists try to, or the pharmaceutical company try to design the drug that can be used to treat some kind of a disease. So the drug screening process they have many terms and many approach. So one of the important approach that in the drug discovery is called the fragment-based screening. So this fragment-based screening is one of the important technique that using the NMR to obtain the new candidates for the treatments. So this is an example to show, okay, from this drug discovery. So we will start from the simple fragment until we get the final drug, or we call the prodrug. And then from the prodrug, so the pharmaceutical company try to develop into the final drug that goes for the clinical trial. So for the drug discovery process, so beside the target that we are looking at, so we also need to know where this drug attack. So it means that very often we also need to understand and identify the target of the drug. So this target of the drug, it can be the RNA, it can be the proteins. Yeah. So for example, here this is a drug, this is a target, and this is we try to perform the fragment-based screening. So if these fragments, they have the special conformations that allow them to interact with the proteins, for example, the targets. So it means that they will enter these pockets with these weak interactions. So by applying some kind of NMR experiments, so we're able to detect, okay, which component that is weakly bind to the targets. So for example, if we have the two fragments, okay, so before we start the measurements, so we will collect the data, NMR data from this free fragment. Then we put these samples into the drugs, into the target protein sample, and then we allow sometimes, okay, so for example, this particular fragments able to interact with these pockets. So when we perform the energy, so for the NMR measurements, so the energy will transfer from the target to the sample. So once this happens, because there's a weak interactions, so when these active fragments, okay, leave the protein targets, so we will measure the NMR for the second time. So by comparing the initial and these during the binding steps of these fragments, so we're able to take the differences. So we're able to get the signal that is actively bind to the target. So whereas the brown color, the fragments which is not bind to the targets are not shown. Okay, so in this fragment-based screening approach, so this is one of the example to show, okay, that these the models, okay. So there have many drugs have been discovered from this fragment-based screening. So vemurafenib, okay, this is one of the example. So it starts from these little fragments, and the pharmaceutical chemistry lab able to develop the full drug, okay, that can be used for the treatments for the cancer. So this is another example. Yeah. So, and of course, beside the classical applications that require the sample to be purified, the applications of NMR now can be extended to the mixture sample. So this mixture sample can obtain from the clinical sample. It can become from the food industry, and it also can be related to the microbiology. So this, I will take an example, so that related to the clinical and the biomedical applications. So the clinical sample usually are from the plasma serums, as Dr. Mongkhol mentioned just now. So of course, you also can collect the saliva, urine, and the cerebrospinal fluid from a human. Yeah. So there are many publications that related to the clinical applications using the NMR spectroscopy. So I will take one of the example that related to the SARS-CoV-2 disease. So this is on the metabolomic approach. So previous parts, the NMR is used to study the RNA and the proteins that come from the SARS-CoV-2 virus. But the approach is on the clinical level. So means that we are not studying the proteins and the RNA on the virus anymore, but we are taking the sample that related to the patients that like either plasma or the urines. So if you take a look into the literature review, so there are many publications that talking about how the NMR can be applied to study the SARS-CoV-2 or the COVID-19 disease by collecting the plasma or the serum from the patients. So this is example showing the plasma sample. Okay, so when you look at the NMR result on the plasma sample, actually it's quite complex. Yeah. So means that in order to fully extract the information out from this plasma sample, very often the scientists need to consider or to perform the analysis using the statistical analysis, or we call the pattern recognition analysis. So the Professor Jeremy Nicholson from Australian National Phenome Center he's a research leader on the SARS-CoV-2 virus study using the metabolomic approach. So there are many publications that come from his lab. So this is one of the examples showing that by analyzing the plasma sample using the NMR spectroscopy and performing some kind of pattern recognitions calculations, so we obtain this type of the graph. So this graph actually is the plot out. It's a plot from the NMR result from the NMR spectrum. So as you can see here, that from the side that's showing the blue one is from the plasma from the healthy sample, and the red color is from the SARS-CoV-2 virus plasma. So when you compare the NMR, so this is the statistical plot. Okay, this is we call the pattern recognition methods. So if you look at the original data, which is the NMR spectrum, so we can actually tell very clear easily. Okay, yeah. So for the healthy samples, okay, and the SARS-CoV-2 virus sample, means that the COVID patients, so they give the very significant change in terms of the plasma profile. So this is example is called the serum profile. Actually, the serum and the plasma is about the same. So from the study indicates that actually for the SARS-CoV-2 patients, it showed some abnormality in terms of the lipid profile. So the SARS-CoV-2 patients demonstrate an abnormal lipoprotein metabolite signature, which is consistent with the coronary artery disease, so which is related to the heart disease. So the contents as the following. So low total and the HDL apolipoproteins A1, low HDL triglyceride and the high LDL and the VLDL triglyceride. So this is a typical patterns from the SARS-CoV-2 patients that can be detected by NMR spectroscopy on the plasma sample. So when you look at this, the lipoprotein, or we call in general the cholesterol. So in general, from the biological lab, okay, or the microbiology lab, the MLT lab, okay, so the cholesterol, it can be roughly divided into 3 categories: high density lipoprotein, low density lipoprotein, and the intermediate density lipoprotein. And actually in NMR, it tells you more about this the lipoprotein profile. So the NMR also can deliver the subclasses beside the main class, okay, beside the high density lipoprotein. So you also can identify all these are the subclasses that related to the high density lipoprotein, and 6 different subclasses from the low density lipoprotein, and 6 different subclasses from the very low density lipoprotein. So this is a very, very specific to the NMR parameter. So it means that by measuring the plasma from the patient, so the NMR result already it can tell us the detail on the subclass analysis. So this is the example to show that how this lipoprotein subclass, okay, related to the plasma and the serum. So we are not just looking at the high density lipoprotein or the low density lipoprotein as a main class, but the NMR also can deliver the information that related to the subfractions. So by looking at these subfractions, so the COVID-19 patients show the very special signature that related to the cardiovascular disease. So this, the lipoprotein subclass analysis, actually the application is very wide. So using the NMR spectroscopy. So one of the example is the COVID-19 disease. So of course, the classical lipoprotein subclass analysis can be applied to study the high blood pressure, fatty liver, Alzheimer, the cardiovascular disease, or even the diabetes. Okay, yeah. So the application related to the lipoprotein subclass derived from NMR is very wide. So of course, the NMR can be applied to the food sample and the nutrition study. So there are also many publications that related to the foodomics, okay, and also the dietary study that using the NMR spectroscopy. So of course, the plants, okay, study using the NMR related to the plant metabolomics also is also very common and very popular. So just before I stop, okay, so for this first topic, so the NMR also can be applied to study, okay, related to the microbiology field. So whether the sample is the in vivo sample or the in situ samples, so both sample can be analyzed by NMR. So when you look at the in vivo sample, so very often the people using the metabolomic approach, okay, to study the chemical constituents from the microbiology. And of course, that can be related to the cell, okay, yeah. So this mammalian cell is just one of the example. Of course, you can use the NMR to study the microbial cell. So you can determine the contents, okay, by performing the quantitative analysis on the cellular the samples, okay. So you can determine the intra and the extracellular metabolism from the microbes as well. So this again, I'm taking the in-cell NMR in human cell. So this also applicable to the in-cell NMR on the microbial cell. Okay, that is also possible. So with this, I finish up my first topic related to the applications of NMR to the various field. So thank you very much. So shall I continue with the second sections?
Yes, please.
Okay.
Thank you.
Okay, So next I will move on to the next topic that related to the QNMR with targeted and non-targeted metabolomics. So I will start again. So I will cover several topics. Okay. So I will start with the quick introductions to NMR parameters and NMR-based metabolomics. So because the metabolomics approach is quite complicated, so for those who don't have any experience in the NMR and they are new in this field, so hopefully by after go through these topics or these little sessions, so at least you get some ideas, the signal or the informations that we can obtain from the NMR. So when you look into the molecular spectroscopy, so there are the several technology, okay, that are related to the molecular spectroscopy, and NMR is one of the molecular spectroscopy technique. So in NMR, so the technique using the electromagnetic radiations. So this is the energy that interact with the sample. So the sample contains a special target. So and when the sample interact with the electromagnetic radiations, so the sample emit the signal. So this signal can be captured by the NMR machine, which is the NMR spectrometer, and we can analyze the signal come from the molecule species. So when you take a look into the details, what the NMR detects. So actually when the electromagnetic radiations will interact with some kind of the target, okay? So this target actually is related to the atoms, okay? So means that the atoms that possess the special features, okay, or the special properties, so that is related to the magnetic properties, so they are NMR sensitive. So those the nuclide with this the magnetogyric ratio, or sometimes people call it gyromagnetic ratio, they are NMR detectable, they are NMR active. So when you take a look into the table, so this is a general table that you can obtain from any website or the NMR textbook. So those the NMR detectable nuclides, so usually they are non-zero spin. So beside we can say with this nuclide gyromagnetic ratio, so you also can say that the NMR active nuclide with the spin quantum number, which is non-zero, not equal to 0. So as long as they are non-zero, they are NMR detectable. Okay. So as I mentioned in the first sessions, so there are a lot of nuclide, they are NMR detectable. So when you look at the favorite NMR nuclide, so the 1H carbon-13 and the nitrogen-15, these are the most popular nuclide in NMR spectroscopy. But compared to these three nuclide, actually the proton 1H is the most favorite nuclide amongst all. Okay, because this nuclide is highly sensitive because of the natural abundance and also the relative nuclear magnetic moment. So many people, when they say they perform the NMR, so most of the time they refer to the 1H proton because this is the most sensitive experiment. So when you look into the very fundamental, so if you have the one HPLC system and you have the sample, and this sample, if this is a mixture, so this sample go through the HPLC system, and usually they need to go through the HPLC column, and then you will go through the separations, and then with the appropriate detector, so you will separate into the components that is related to the sample. So if you have the 3 components in the mixture, so you will separate into 3 peaks, okay? So which is we call the chromatograms. So it means that the 1 peaks refer to 1 component. So the second peak is referred to another component, and the third peak refer to the third component. So when you take a look into the NMR NMR result, so if you have the sample that contains 1 component, so are you getting 1 peaks, okay, from the NMR data? So this is the one questions that you need to ask. Okay, so if you don't have any experience, so you say yes from the HPLC, 1 peaks correspond to 1 component. So but when you take a more details check, okay, on the NMR, so actually we are not getting a peak, okay, but we are getting a group of signals. So this result actually is obtained in 1950s on the samples that is called the alcohol ethanol. So when this is the result from the 1950s. So if you have the NMR machine in your lab, okay, so you easily obtain this type of result, like for example, the 1D and the 2D NMR spectrums, okay, or the NMR spectra. So this the NMR signals, okay. So for example, if you have the very simple molecules, so when you measure with the NMR, so we are getting a signal. So this is one signal, yeah, that belongs to this molecule. So that is very similar to the HPLC. So one compound, one signal. So this is another example showing here. So you're getting the blue signal. Okay, so single signal. Okay, that is belong to this compound. So and this is another molecule, another sample also giving you a single signal. Okay, then you will ask, okay, yeah. So is it all the molecules always give you one signal? So the answer is no. So as the molecules become slightly complex, so you have more groups of signal. So for example, this molecule, you have the red color signal. So this is another molecule that give you the orange color, the signal that belongs to this molecule. And as the molecules become more and more complex, so this is from the purple, so that it belongs to this molecule. And the signal complexity, it go according to the molecular complexity. So if you have the more and more complex molecule, so you expect more and more signal that is come from the same molecule. So it means that in most of the time, we are not getting a peak for a molecule. So usually we are getting multiple signal groups, okay? Multiple signal group. So for example, this molecule, so all these signal are belongs, green color signal are belong to this molecule. Yeah, even though it's very small molecule. So this is another example. Yeah, so another example. So as the molecular complexity increases, so the spectrums of the NMR, okay, become more and more complex. So as you can see here, this is the one of the example of the sugar. So it gives us so many peaks, okay. So this is the group. This is another types of sugar, and this is another types of sugar. So the what I try to point out is actually the NMR signals, okay, is very useful, and this is compound specific. By analyzing the spectrums, so we can tell what types of molecules that this signal belongs to. So it's a compound specific, and it tells us about the chemical fingerprint of the compound. And the NMR result is very reliable. Okay, so when you take a look, okay, so on the upper trace, this is the spectrums that come from the mixture. Okay, so if you can have the way, so actually this signal from the mixture actually is produced, okay, or generate from the individual contents that form the mixture. So if you have 10 components in the mixture, so these 10 individual chemical components, individually they'll produce their signal, okay, at the certain range, and then they'll combine everything from these 10 components into the final spectrum that you obtain when you measure the mixture. Okay, when you measure the mixture, so it means that the signal from the NMR from the individual component will be co-added. Okay, so it means that we are obtaining the total result that can be further break down into the individual component if you have the way to trace back. Yeah. So as I said, the NMR result not just highly compound specific, but the result is also very, very reliable. So and because they will give us very reproducible result. So with this, the compound specific and the reproducible result. So in the mixture analysis or the field that related to the metabolomics, so very often people talk about the database. So by comparing the mixture data with the library that they can access to, or we call the spectral database they can access to, so they're able to identify some of the component. So later I will talk about this, okay, yeah, in the next few section. So when I take a look at the NMR result, so which is compound specific and the chemical fingerprint. So different compound, they will give you the different fingerprints, and the signals will spread along the axis, so which is known as a chemical shift axis. So this is the chemical shift axis, and the the level, or we call the unit is around ppm. So when you take a look at this particular molecule, yeah, so this is the molecule that gives the signal on the red color traces. So the molecules contains the different atom connectivities, okay? So this is forming this molecule. So the different locations in the molecules will give the signal at a certain position. So means that when you have the molecule with the different connectivity, so every positions, okay, in the molecule will give a corresponding position signal in NMR. So it means that the NMR actually is a very powerful tool to probe the chemical environment within the molecule. So this is for one molecule. So of course, if you have another molecule with the different connectivity, so by analyzing the signal, so actually we can tell which signals which signal come from the molecule. So it means that when you take a look at these positions in the NMR, which is we call the chemical shift. So analyzing the chemical shift allows us to understand the functional group, which is the link to the molecular connectivity level. So that is very, very important, especially when you try to analyze on the mixture sample. So on top of that, okay, so we are not just having the signal that spread along the different positions, or we call the chemical shift. So when you look at the NMR result, so which is the pattern, okay, so you can observe sometimes some sample, it give you one single peak, okay, one single peak. But very often that NMR result, you are getting more complicated pattern. So which is commonly known as splitting pattern. So how this splitting pattern arise, okay? So instead of getting one single peak, but we are getting multiple peaks, okay, per group, multiple peaks per group. So actually, this have to be, we can understand this from the molecular level. So we know that NMR detect all the nuclei that have the magnetic properties, so which is the gyromagnetic ratio. So for example, this group, okay, the methylene group, okay, so you have the signal here, and the methyl group, because of the chemical environments that are different, so their signal will resonate at the different frequencies, okay, so this I already mentioned, this is a chemical shift, and they are magnet. And because these are the tiny magnet that able to, they are joined through covalent bond, and this covalent bond actually is very short. So with this tiny magnet that join through the covalent bond, so these magnets able to interact between each other. So when the magnet interactions happen, so it will cause the signal, instead of single peak, to become the multiple peak. So this is actually the very fundamental in the NMR spectroscopy. So which is the involve the covalent bonds, okay, and the magnet interactions within the molecule will cause the signal to be splitted. So this is the result from the covalent bond and the magnet interaction, so which is called spin-spin coupling. So when you look at the NMR result, so very often we are getting this type of patterns, okay? Instead of single peak, we are getting multiple peak per group. And when you try to identify, okay, the signal that belongs to the particular, come from the particular chemical constituent, so we are not just analyzing the position, but we also looking at the pattern, okay, or the splitting pattern or the multiplets, so that we use this as the confirmation steps, okay. Yeah, so it means that we have the informations come from the where the signal are located, so which is a chemical shift. And at the same time, in addition to that, we're also looking at the pattern. So the pattern will tell us whether we are looking at the same compound or not. So the third information that also obtains easily from the NMR result is the peaks, okay? That, for example, here, the intensity, yeah. So this is a high intensity, this is a low intensity. So this intensity, or we call the area under the curve, or we call the integral in NMR. So actually it tells us about the number of atoms. So for example, if we have the 3H in the methyl group, so this 3 hydrogen will give you the tall peak. Whereas if you only have one H attached to the methyl group, so the signal is only 1, Okay, so it means that actually the area under the curve or the intensity of the NMR signal, it go together with the atoms, the number of atoms. If you have more atoms, it means that you have taller intensity. So this is very important in NMR analysis, okay? So if you have the topic, so for example, the area under the curve showing here is a 3, and the area under the curve showing here is 2. So it means that we can trace back to the molecular level which atoms, which group that contribute to this area of 3. So which is, of course, this is a methyl group. And the methylene groups, which is the CH2, will contribute to the area of 2. So the number of hydrogen atoms is proportional to the area. And this is very, very important because the quantitative NMR will rely on the area under the curve. So if the sample is pure, you know the information about the number of atoms. When the sample is in the mixture form, the area under the curve will tell you the concentrations of the sample in the mixture. Okay, so this concentrations is related when the sample is in the mixture form. So when you take a look into the mixture analysis, that is also known as the metabolomics. Okay, so we are studying the mixture from the varieties of sample, from the clinical sample, from the plant, from the pharmaceutical study, from the microbiology, from the in-cell research. Okay, multiple source. Okay, so what are the key, okay, to the success of the metabolomic study? So first of all, you need to prepare a good sample. You need to have the good SOP to prepare the sample as well. So on top of that, you need to have the NMR machine that have always tuned up to the best, okay, optimum machine performance. And you also need to have the good way to acquire your data, okay, before you have the NMR result that can be used for the analysis. So all these are the three important steps, okay, prior to the good NMR data. So as Dr. Mongkhol mentions that we will try to prepare the sample as shortly as possible, okay? Yeah, so because we do not know whether the sample may degrade or not. And the machine performance have to be checked, okay? So for example, you need to make sure that your machine is properly temperature calibrated because the temperature may shift the signal positions. So you need to make sure that your shim performance is also checked, okay? Yeah, because the shim will make sure that you have the very narrow and the sharp peak. And of course, the machine calibrations need to be performed also on the calibration has to be performed so that you are measuring your sample after the sample have reached the thermal equilibrium. Okay, so we do not want to measure the sample while the temperature is not yet stable. And during the measurements, okay, yeah, so we try to perform the pulse calibrations on each of the NMR sample so that you have the best sensitivity on every sample. So as I mentioned that there are many steps that you need to take care, okay, especially when you work on the mixture, because the mixture analysis require the high. You need the NMR spectrometer to be highly performed. High performance machine will deliver the good result. So the mixture analysis is one of the study that require the high performance from the NMR spectrometer. So after you get all this done, then come to the data analysis, okay? So the data analysis can be performed on the two different software that we offer. So one is the actual NMR and another software is called Amix, okay? So when you look into the study or the data analysis on the mixture sample, that is related to the extract as well, yeah? So there are two approaches that they are commonly applied. So one is known as the replications, another is commonly known as the metabolomics. So when you take a closer look on these, the dereplications and the metabolomics, so the dereplications, sometimes people call it targeted screening. So this targeted screening, it means that we will try to analyze our mixture spectrums by comparing the sample, which is from the mixture, with the library, which is the known compound. So we will try to match the signal, the position, the chemical shift and also we try to match the splitting pattern with the library. So after we perform this compound identification, which is a match, then we perform the quantifications. So this will be the subsequent steps from this targeted screening, or sometimes people call it dereplication. So sometimes people call it metabolic profiling. Okay, metabolic profiling. So another important approach that is also commonly applied to the mixture analysis is called metabolomics. So sometimes people call it the non-targeted screening or the untargeted screening, or sometimes people call it chemometric analysis. So basically, this is the step that using the mathematical calculations, okay, to perform the fingerprints analysis. So usually we are putting the whole NMR spectrum into the statistical software to perform this chemometric analysis. So sometimes people call it multivariate analysis, or sometimes people call it metabolic fingerprinting. So I will start the talks that related to the metabolic profiling. So involve the two components, identifications and quantifications. So the identifications can be performed by comparing the NMR result from the mixture with the library. So the component, okay, in the mixture, the sample from the mixture, it can be, the sample can come from it can contains the primary metabolite or the primary markers, or for example in the plant sample, so very often that there is also the secondary metabolites that present in the plant sample. So that is called the secondary marker. So whether it's a primary marker or the secondary marker, so you need something to compare. So you need a library, you need a database. So the comparisons, the spectral comparisons with the library is one important step. But before you can perform the identifications, make sure that you compare your sample, so which is the mixture, with the library under the similar conditions. So it means that we make sure that the pH that you compare between the sample and the library are the same, buffer between the sample and the library are the same, the solvent are the same, the temperature measure on your sample and the library are the same, and also the magnetic field. Because once you change one of these parameter, the positions of the signal will may be shifted, then you are not able to compare directly. So this is an example to show that the same sample, but they dissolve in the different solvent. So the chemical shift or the positions of the signals are different. So the pH also have the important effect to the chemical shift. So it means that you need to make sure that you are comparing the same pH. So the library that I'm using for this study, I'm using the Bruker BioFluid Reference Compound Database, so which in short is called the BBioRefCode. So in this library, it contains 850 compounds, and all these compounds are purchased and measured in our application labs. So these are the pure compound spectrum. So pure compound spectra. So they are not synthetic. They are not synthetic. So they are real NMR spectrum. And the sample are measured. All the sample are measured in the buffer and in 600 megahertz. So they have also contains, this library also contains the 2D data. So and the three different pH 3, 5, and 7. Of course, for the full packages, the pH will the pH of the NMR library start from pH 3 until pH 8 at every 0.5 pH jumps. So this is the BBioRefCode that I'm using for the comparisons. So for the identifications, so that the sample is on the energy drink, but I'm using the BBioRefCode for the identifications. Okay. So these samples actually is measured by Dr.Mongkhol. So he's using the SWU NMR system. Okay, yeah. So thanks for the university that allow us to measure this energy drink. So we have energy drink come from the different brands. Okay, And this is the example to show that from the different brands. Okay, yeah. So I didn't highlight. I didn't hide this brand. I just put the brand here, but just to comparison. So this slide just to want to show that the pH. So we have the 3 different pH. So this is a pH 3.5. So this is the pH 4. And then this is a pH 4.5. Okay, we have the 3 different pH. So we try to compare from the NMR fingerprints. Okay, how sensitive the pH. Okay, yeah, the effect of the pH on the NMR sample. So first of all, from the pH 3, okay, which is in the orange color. So the sample was measured by using the pH meter. So the pH meter reads at 2.9. So and the pH 3 is very close. So means that the result is very they are in good agreement. So for the next sample, this sample in the green. So the pH is from the NMR estimated is around 3, okay, or less than 3.5, okay, and the pH meter showing that this is 3.1. And for the blue one, so the pH meter showing as a 3.1, but the NMR showing there's some kind of drift. So as you can see here, that as the pH move to the higher value, which is to the more neutral value, so the peak will shift from left to right. So the pH meter cannot really tell the peak, okay, the pH on the green, and there's a slightly different between the green and the blue, but the NMR can tell from the peak, okay, the position of the peak. So then we move on. So for the pH, this is close to the pH 2. This is a 3.1. This is a pH 3. This is a pH 3. This is a pH 3.2. So the signal already moved to 3.2. That is also good agreement. And then here the signal shift a little bit here to the right. Okay, so the pH showing higher. So that is also quite good agreement. So, but when you compare these two, also the positions of the brown and the greens, okay, they are about the same. So the pH meter showing the 3.4, 3.4. So they are good. Okay. So we have one sample that is belongs to the purple group. Okay. So the pH meter showing the 3.6. Okay. But from the NMR spectrum, it shows the signal actually is less than the 3.5. Okay. So this is the slightly different between the NMR result and the pH meter. So another sample that come from the color in the gray. Okay. So the pH meter showing the 3.7. So, but in the NMR, it show close to the 3.5. Okay, so this again slightly difference between the pH meter and the NMR result. So this is the signal that come from the orange is already moved beyond 3.5. Okay, which is the purple line. Okay, so which is also from the pH meter showing the 3.7. So yes, again this is good. And the last one is the black color. So as is shown in this, the pH meter showing the 3.9. Okay, but here, yeah, the from the NMR seems not really correct, because the pH this is a 3.5. This is a pH 4. So it's already actually is then this is a 4.5. Okay, so it's slightly moved to this point. So means that maybe this is not very correct. So probably the measurements of the pH need to be redone. Okay, because we know that the NMR is very sensitive. So this is just to show to you that actually the pH have the important effect on the NMR result. Okay, on the NMR result. So again, here this is just to compare in general. So we have the different types of energy drink. Okay, so actually the result, despite the pH, the sensitive pH sensitive, so in general the results looks well, looks good. So for the black sample, the black coded, blue coded, green coded come from the same types of energy drink. So actually the patterns or the chemical profiles looks very consistent. So for the repeats. So these are the expansion regions. So you see here the black, the blue, the green from the different brands, okay, of the energy drink. So they are very, very consistent. Okay. So here this is another expansions regions. Okay, this is another expansion region. So here just to indicate that the patterns or the chemical fingerprints are very, very consistent that related to the particular sample types. Okay. So when you try to do the further analysis on this energy drink, so actually I take one of the representative sample. Okay, so I try to analyze with the actual NMR software, and I try to identify from the library. So again, so the pH have to be defined. Okay, so this sample I checked already. The pH is around 3.5. So the identification is performed on the 3.5. So as you can see here, at the top is the mixture from the energy drink. So we try to compare the fingerprints from the different component. So the component brown, component purple that correspond to the certain metabolite. So one by one, so we can try to match the positions, and we can try to match the splitting patterns. So here I already mentioned just now that we are not just looking at the signal positions, which is the chemical shift. So we also can make use of the patterns, the splitting pattern. So from this the database, so actually we can try to call up one by one, and we can manually compare the signal pattern. As you can see here, yeah. So this compound, this pattern is very similar to the mixture, which is the black, which is the energy drink. So we can one by one we identify from the library by matching the chemical fingerprint. So this is we known as the replications, or sometimes we call it the metabolic profiling. So the profiling need to be you need to perform the profiling using the similar sample concentrations with the library. So it means that I'm using the pH 3.5 for the metabolic profiling. So it means that the one by one, so we can compare the fingerprint, and we try to match we try to match the splitting pattern and as well as the chemical shift. So with this approach, so we can identify a number of compound from the library based on the chemical shift and based on the splitting patterns. So of course, we will expect some kind of a shift between the sample and the mix and the library, because the sample is the pure component. Okay, the library is a pure component, and the sample is a mixture. So we will expect also some kind of the molecular interaction in the sample that cause the signal position to be slightly shifted a little bit. So here, this is the area that is overlap of the multiple group of signal. So as you can see here, that when you look at the patterns here, the patterns showing that they are non-symmetry. So when they are non-symmetry, very often there's an overlap around. There's an overlap of the signal, a group of signal here. So when you have the overlapping, so it means that even there's overlapping, but you can identify it easily by comparing the pure compound library with the mixture. So when you overlay, then this pattern can be easily recognized. Okay, yeah. So by comparing this fingerprint one by one, so we are doing the identification from the library. So as you can see here, that the big shift, yeah, as I said, yeah, this is expected because the sample is a mixture and the library is a pure compound. So we don't expect the molecular interactions between the different types of molecule in the pure sample, because you only have single types. Yeah. So this is important, okay, in the metabolic profiling. So we can overlay our sample with the library, and we can move the spectrum up and down, and we perform these identification steps. So after this, the manual step identifications, so there are number of compounds that have been successfully identified. So this is the one part of the spectrums, which is the chemical shift more than 6.5 ppm. So this is the regions that is related to the sugar area. So the identifications was performed, and these are the signals that have been successfully identified. And this is the expansion regions. So on the small components that presents together with the huge contents. Okay. And this is the regions that is the area start from the 3.0 and before. Okay. And this is another last part of the spectrum. Okay. That related to the overlapping regions that I already shown in the video. So it means that with this, they'll be able to identify a number of compounds. So usually after we successfully identify the compound signal locations, which is the chemical shift, so the next step is to try to perform the quantitative analysis on that mixture. So here, I'm using one of the options, okay, so to perform the area under the curve. So I'm using this option. Of course, there are several options that you can use. So this option is one of the options that the Bruker spin allow. So on the certain regions, okay, so I try to integrate the signal. So one of the signals that I try to point out, okay, yeah, so highlighted in the red color here, yeah, this signal. So you see that this signal is a part of the mixture, and we have the very intense peaks, okay, or the intense signal, and this signal is considered not very intense, okay, yeah, so this is intense signal, this is not very intense. So when you try to do the quantifications on the less intense signal, or we say the minute compound, okay, or the minute component. So you need to make sure that this minute component still give you sufficient sensitivity. So we can determine the sensitivity by performing a step called the signal count. So in this case, this signal with this signal count 88.73. So from our experience, so any signal value less than 150, so you're expecting higher error. Okay, so we will try to perform the quantitative analysis on the NMR result with the signal value more than 150. And of course, the higher the better. The higher the value, the better the accuracy, or the better the precisions. So it means that in this case, the signal that is close to this level is suffer from the accuracy and the precisions. So of course, you still can perform the quantitative analysis on this particular peak, but you are expecting higher error, okay, higher error. So in the quantitative analysis, so we need to understand the spectrum as well. Yeah, so we know that this is the mixture, okay, this is from the one of the example energy drink. So and we managed to identify the compound that related to the signal. And in the quantitative analysis, we are not just identified from the compound, but we need to know from which group that this signal belongs to, so that we can tell how many proton that correspond to this group of signal. So for example, this molecule, okay, this is a niacinamide. So this signal is correspond to this CH, okay, in this functional group. So means that this area under the curve, okay, for this compound actually is contributed by 1 proton. And with the concentration, so this is the area under the curve. Okay, so hopefully that this is clear to you. Yeah. So for example, if you have the second molecule that is called the benzoic acid, so this benzoic acid contains multiple signal. Okay, multiple signal which is highlighted in green. So for the quantitative analysis, so actually we just need at least one area that correspond to the molecule. So the rest, of course we can take the average, but at least we need to have the one area that can be linked directly to the compound, and we know that very well that this area is due to this particular group, which is the 1H. So in this, the third example showing that this is the para-anisic acid. Okay, so that they have multiple signal, but we need to identify at least one per group per molecule. So this group of signal actually is contributed by 2 protons. Okay, 2 protons. So, and then the last example in this slide is showing that this is the signal that belongs to this 2CH. So this signal is a single peak, and this is belong to the 2H, 2 proton, 1 CH here and 1 CH here. So you need to know from the area under the curve, which part of this area refer to the molecule and how many proton that correspond to this area. So with these informations, then we can calculate, or we can do the quantitative analysis on the mixture. So similar approach applied to the rest of the molecule. So for example, this molecule, this area under the curve, so this is the area, and then this is belong to the 1H. And we have 3 more molecules here. These are the different types of sugars. Okay, sucrose. This is the anomeric proton resonates at this frequency. Okay, and this is belong to 1H. And this is the glucose. Okay, this is the anomeric proton. So which is highlighted in the red color. So you may ask me why the 1 proton here. Yeah. So in the glucose sample, and you have 2 different areas. So actually, this glucose molecule can undergo change between the alpha and the beta glucose form. Okay, so there's the equilibrium between the alpha glucose and the beta glucose. So this is the alpha glucose, this is the beta glucose signal. So that's why you have 2 different areas. And for the malic acid, okay, so which is highlighted in the pink. Okay, so this is the molecule that is correspond to 1H. So another molecule, silico silo inositol. So these molecules, these hydrogens, okay, 6 hydrogens will give to the signal that is showing here in this highlighted in orange color. So with the same approach, so we are able to identify the area, and we can tell how many proton per group, okay, within that can link to the molecule. So this is the another a molecule called the propyl acetates, okay, which is also commonly found in the food samples. So this is a CH3 that correspond to this signal. So this is a 3 hydrogens per this group of signal. And finally, the molecule lactic acid, that is another CH3, and then this will give rise to the area under the curve here, okay, that is belongs to the 3H. So by understand the area under the curve that belongs to the molecule, and also the number of atoms that correspond to each area. So at least 1 molecule, we need to have 1 area. Then with this information, we can perform the quantitative analysis on every molecule. So at least you need to have 1 area, specific area for that molecule that is not overlapped with other, so that the signal can is isolated, or the baseline separation. So we can use that signal for the quantifications of that chemical constituents. So I hope that this is clear to you. Yeah. So when you look into some basic fundamental that related to the quantitative NMR, so actually NMR is defined as the primary methods. Okay, so there are many primary methods available. Okay, yeah. So NMR is one of the primary methods, and by definition, actually NMR is a primary ratio method, and is also known as a relative method. So when you look into this primary ratio method and the relative method by definition, so the primary ratio method measures the ratios of related measurements. For example, the purity of the pure compound and the amount of substances in the mixture. So you can use it for the determinations of the purity of the compound or the amount in the mixture by comparing the signal of this sample with the standard. So it means that we need to compare with the standard. So we can compare with any standard with the known compositions. Okay, so this standard in NMR is commonly known as certified reference material. Okay, so another background that related to the quantitative NMR spectroscopy. So the area under the curve from the NMR actually is based on the structural property. So this is very specific to NMR, structural property. So some other techniques, okay, that allow the quantifications. So those techniques very often they based on the physical property, but NMR is based on the structural property, not physical property. So when the technique is based on the structural property, the technique is known as universal. So when the technique is based on the physical property, then this physical property means that the methods are very selective. So sometimes people call it wide selectivity to explain the universality of the NMR technique. So why this is important? Okay, so the structural property means that if you have the CH3, so the area under the curve will be 3. If you have the CH, the area under the curve will be 1. Okay, and if you have the CH here, and then the area under the curve will be 1. So this is we call the structural property. Means that if the structure contains the 3 atoms, then the area will be 3. If the structure contains 1 atom, then area will be 1. So this is we call the structural property. So because the NMR is based on the structural property, So means that in order to identify or in order to quantify 3 components in the mixture, so and the NMR is a universal, universal because it's based on the structure property. So in order to quantify the component in the mixture, so unlike the classical method or unlike other analytical methods that you need the calibration curve, so for NMR, because it's based on the structural property, so you do not need the calibration curve, and you do not need any curve, okay, to determine the response factor. So, and you just need a certified reference material with known concentration, and you can take any certified reference material. And related to this point, you use the non-structural identical calibrant. You can use any, okay, any CRM to quantify all these 3 components. Whereas, as opposed to other method that related to the physical property, you need to plot the calibration curve. You need to determine the response factor, and of course, you need to choose the identical calibrant for the calculation, for the quantification. So NMR, you do not need that because NMR is the universal method. So you just need any CRM, okay? And in NMR, you do not need a calibration curve, you just need a single point. And this single point will allow you to perform the multiple analyte quantification in one shot. So whereas in other method, you need to have the several calibration curve in order to determine those quantification, in order to determine the quantity of the different component. Yeah. So because NMR is using the single point, so we have 2 options. So depends on the methods that you are using, whether you are using the internal referencing method, which is using the orange equation, and if you're using the external referencing method, then you are using this blue equation. So this will depend on the method that you apply, okay, for the quantitative NMR analysis. So the NMR can provide us the absolute quantification. So as long as you can calibrate your NMR signal with the known certified reference material, known concentration, so you already can establish the absolute quantification. And the beauty of this absolute quantifications, or the advantage of this absolute quantification, is as the following. So the purity of the major component can be evaluated regardless of whether other components are observable or NMR silence. So it means that even though the components impurity or the content, they are non-NMR detectable, doesn't matter because we calculate the sample purity based on the weights, based on the mass. So as long as you know the total weight, means that of the mixture, okay, then you can determine the gram percent of the sample. So that is very, very important. So that is the beauty of this absolute quantification. So this absolute method cannot be applied to the mixture unless at least one specific proton signal can be assigned to each of the components within the mixture. And the number of the proton of the signal is known. So as I already mentioned to you, so as long as you can tell the particular signal, specific signal that corresponds to that chemicals, and you know the number of proton, you can do the quantifications on that component in the mixture. So as I said, there are 2 different ways that we can perform the quantifications using NMR. So one is the internal, one is the external. And internal means that we add the standard material into the sample that we want to analyze. So it means that they are present in the same NMR tube. So when you measure the sample, so you have the signal spectrum, the component that related to the standard and the component that related to the sample that you want to analyze. So this is the internal method. So of course, you can have another way, so which is the external method. So we measure the reference material in the separate NMR tube from the sample. So it means that now we measure 2 samples. So one is the reference material that we use this to calibrate our signal, and another sample which is the analyze. Yeah. So two two different tubes. So this is the external reference method, and this is the internal referencing method. So yeah, in this energy drink, so we will focus on the external standard method. So first, the sample is on the pure reference sample. So this sample actually is our sucrose sample. Okay, so our Bruker standard sucrose sample. So the concentrations of the sucrose is already known. So it's the 2 millimolar, and the sample is in the 600 microliter or the 0.6 ml. So we have the first informations that we related to the standard sample, the concentration. So we know the area under the curve, okay, where is the signal locations, and we know how many proton that these signals belongs to. So we can calculate the area that related to this signal to this proton. Means that yeah, this is a proton. And we also know the molecular weight of this reference material. So we can plug in the informations that we know that is highlighted in green, okay? And we define it as the reference. So this is the external reference. So it means that the external reference also means that we have another NMR tube that contains the NMR sample, which is the energy drink. So in this energy drink, so as prepared by Dr. Mongkhol, so we dissolve the sample, and the final volume is 600 microliter, okay? So which is the 0.6 ml. And I already identified those the area under the curve signal that belongs to the metabolites. So these are the metabolites, okay? So it means that we can set up one by one the area under the curve that belongs to this niacinamide, and then this is the molecular mass, and then this is the benzoic acid. So the area under the curve, then this is belongs to 1 proton and the molecular weights. And the third one is the para-anistic acid. So one by one, we set up this table, and with the corresponding area and also the corresponding number of the atom that contribute to the area, and together with the molar mass. So we can calculate using the top spin by applying the external method, applying this erratic tool, apply on the equations PUCON. So the PUCON is, this is the PUCON. This is the equation that is embedded in the top spin, and we're able to get this type of the result. So the software will calculate based on the informations that we add, we key in, and these are the concentrations in the millimolar. Of course, it also give us the the mass amount, okay, from the calculations. So this is from the different part of the spectra, okay. So I put into the table. So this energy drink, okay, I try to quantify the important components that belongs to the energy drink. So these are the concentrations after the calculations, okay. So as you can see here, that from the first sample from the brand 1, okay, brand number 1. So actually the main content from the energy drink, the main content. So as you can see here, there is the sucrose, okay, yeah, sucrose. So showing here in the sample 1, for the brand 1 is a 219.88 millimolar. So the second major component is the glucose, okay, the glucose. So because I said that there's two anomeric proton from the alpha glucose and the beta glucose, so the content for the glucose we can add it up, okay. So this is the second component, the major component. And then of course we also found there are some other the additional, for example, the acids and the alcohols or the ester that is also present together. Okay, so these are the calculations result. So I try to perform the similar analysis on another sample. Okay, so this is the sample number 2. Okay, and then I found that actually this is the replicates. Okay, so we prepare from the same brand 2 samples. Okay, so as replicates. So the the preliminary calculations shows that yeah, the results are quite consistent. Okay, yeah. So these are the individual chemicals. Okay, and the respective concentrations. Okay, so means that the results are quite close to each other. Okay, yeah, between the two different replicates. Okay, on the same brands. So the majority is the sucrose. Okay. So, or maybe sometimes in the label, they just put it as sugar, okay? And then we have the glucose, okay? Yeah. So energy drink, yeah, it contain high content of sucrose, so it is not good for health, okay? Yeah, so if you have the diabetes, so maybe you try to reduce the energy drink consumption. Yeah. So next, I will move on to the metabolic fingerprinting, okay? So this metabolic fingerprinting required the pattern recognition, so which is a statistical analysis that apply to the NMR data. So sometimes people call it the chemometric analysis or the multivariate analysis. So there are a lot of variety or the mathematic approach that you can apply. So for example, the PCA, PRSDA, yeah, OPRSDA, yeah, so a different versions of the statistic that you can perform on the NMR data. And the final result that you obtain is some sort like this. Okay, this is a statistical plot. So this statistical plot actually is the outcome after we perform the statistical analysis or we perform the multivariate analysis. So how can we obtain this type of statistical result? So actually this statistical result is come from the number, or we call the table with a lot of number. So in the mathematics, so we call it the bucket table. Okay, so we have this plot as a final output, but this plot actually is come from the number. So all the mathematics software only can handle the number. Okay, yeah. So we need to provide the number to the software so that the software can calculate based on the number to get us the plots. So this number, the table that contain the number is important. So in NMR, so we call it the bucket table. Okay, bucket table. And how this bucket table are how can we obtain this bucket table or the table of number? So actually it's come from the procedure called the bucketing. So we have the steps of the bucketing that convert the something to the number, and we need to perform the bucketing methods, okay, in order to get the number. And this the bucketing step is applied on NMR result. So it means that if you have the NMR result, if you perform the bucketing process, you will get the number. So after you have the number, then you can use the software to do the statistical analysis. So this is the short. This is in short. Okay. So this bucketing. Yeah. So what is bucketing? Bucketing means that we'll try to take a cut into the NMR result. So we try to slice the NMR spectrum into the small component. So this is we call the bucketing. So it means that if you have the number of sample, let's say the different types of the energy drink. So let's say 10 sample or 100 sample. So this in NMR, in statistics, they call observation. So we are going to cut the NMR spectrum into the slices. Okay. So this is we call the bucket. Okay, buckets, bucketing. Okay, bucketing of buckets, and you have the result. Okay, which is the table, number of the table. So this is the bucket table, and from the number, the table that contain the number, then we can perform the statistic. Okay. So if you do the statistic on the only on the certain number, so let's say if you are interested on this particular area, then this is we call the unit variate analysis per region. So if you want to analyze everything, means that you are interested on the many regions, many buckets, many components, many variables. So we are doing the multivariate analysis. So to make it simple, yeah. So let's say if you have the sample that come from the honey, and this is from the honey A, you collect the NMR result, NMR spectrum from honey A. And let's say you have the brand honey B, so you collect the NMR result. So by performing the statistical analysis, what do you get? So if you, for example, if you have 6 sample, so 6 sample, and you collect 6 NMR data. So after you perform this statistical analysis, so you're going to have 1 spectrum and 1 statistical dot. And if you have 6 sample, so you're going to have 6 statistical dots, okay? So it means that this is very, very important. So it means that how many sample that you have, so you're going to obtain how many dots in the statistical plot, okay? So this is, for example, on the honey A. So of course we have the honey B. So the honey B also 6 samples. So it means that you expect to obtain the 6 different point from the statistical analysis. So that is belongs to the honey bee. So when you have the overall plot that they belong to the honey A and the honey B, so this is the statistical plot that you obtain. So sometimes we call the mathematic model, okay, mathematic model. So this is a result, okay. So from the energy drink, so I perform the statistical analysis, I perform the bucketing, and then I perform the statistical analysis. So as you can see here that the different brands, okay, they can give the very good clustering, the grouping. So this is based on the brand. So we do have the different bottles, okay, bottle 1 and bottle 2, and then we measure. So you see that, okay, yeah, in the reproducibility, yeah, it shows that here the sample, here some kind of the grouping between the bottles 1 and the bottles 2, okay, for this brand, the GSD and the Lipo Plus, okay, but the rest are quite close, okay, between the bottle 1 and the bottle 2s. So we also do some kind of repetitions on the NMR measurement. So this is the repetitions. So actually the NMR repetitions means that the results are very consistent and very reproducible. So we'll take this example and then we try to see, yeah, the group between this brand and this brand. So from the statistical analysis, it shows that there are 2 groups, okay, 2 groups. So we further take another statistics, so which is we call the SIM count, okay. So we know that there are 2 groups, okay. So we want to test how confidence, okay, yeah, between these 2 groups. So we perform the statistical analysis called the SIM count. So we take the Red Bull as the model, okay? So which is all the Red Bull here, and then we take the Red Bull Extra as a sample. So as you can see here that this is a Red Bull model, all in the green, and this is a Red Bull Extra. So indicates that the Red Bull Extra is very different from the Red Bull sample. So this is one example to show how the additional statistical analysis can be performed in order to understand this distribution. So it means that they are really different. So similar, so we try to see this between the Red Bull sample and the M150. Okay, so from this statistical plot, they are very far. So we want to double check. So we take the same approach. We will build the model from the Red Bull, and then we take the M150 as a sample. So as you can see here, all these 150 are in the red color, means that they are very different from the Red Bull. Okay, yeah. So then because these 2 GSD and the Lipo Plus, they are showing the very special separations. So we want to see whether we can use the same approach to tell whether they are the same or not the same. So first, we will start with the Lipo Plus as the models, okay? So we will take this as the model, and then we take the GSD as the sample. So again, this SIM card analysis showing that the GSD is very different from the Lipo Plus. So similar approach apply. So we can take this, the GSD as the model and the Lipo Plus as the sample. So this is the GSD as the model and the Lipo Plus as the samples. So it's also showing that even those in the statistics, okay, they are very far, okay, between the bottles 1, bottle 2s, but the GSD sample and the Lipo Plus sample, they are very different, okay, they are very different. So of course, the statistic can be performed also on the calculations by excluding the sugar. So we know that the sugar is the main constituents in the energy drink. So we also can perform the statistic by excluding all the sugar, okay, in the calculation. So this is the PCA plot. So when you compare the statistical analysis between the sample that we exclude the sugar, okay, from the calculations, and the sample that without excluding the sugar, so it shows that actually the cluster, yeah, they are still very far, okay, yeah. So probably some detailed analysis need to be done on the excluding the sugar parts, okay, yeah. So this is just to compare the preliminary study, okay, just to compare the overall result without exclude the sugar and the result that exclude the sugar. Yeah. So there's also a number of the informations that you can obtain from the internet. So of course, you can get to these webs, okay? So the NMR applications webinars, okay, that talk about the quantitative NMR and the metabolomics in details. And of course, we also record a number of the webinars, okay, from our customers, okay? So if you're interested, you can also join this webinar on demand. And we also have a number of resources that related to the metabolomics. Yeah. So we also conducted a number of the class, okay, NMR training class, okay, by our colleagues. So from the Bruker United States, okay, so this is related to the mixture analysis, okay, on the complex mixture. So if you have time, maybe you can go to join this training on demand. Yeah. So just before I stop, so I would like to thank our colleague from Germany, Switzerland, and Bruker for the quantitative NMR contributions, and also for the metabolomics part. Okay, the Bruker Germany colleagues and Bruker and the Bruker Japan colleague for the contribution of the metabolomics NMR in general. Yeah. So with this, I finish up my talk, and I would like to thank every one of you for your attention. Thank you.